Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A Biology Unit 2, potential questions that could come in any Unit 2 biology paper. Let's begin with Topic 3. Topic 3, Cell Structure, Reproduction and Development. Here I began by looking at microscopes. They could ask you about the light microscope and the electron microscope in terms of the advantages as well as disadvantages. They could give you images about the scanning electron microscope and the transmission electron microscope and ask you for the differences in terms of magnification, resolution, as well as if they are 3D or 2D. Images can be given and they can ask you whether they are taken using the scanning electron microscope or the transmission electron microscope, so please ensure you fully observe the image before you make a conclusion. The next part is the cell and organelles. You need to learn to draw an animal cell and label the inner components like the cytoplasm, the nucleus, and all the other organelles. You need to remember the functions of organelles like the mitochondria, the Golgi apparatus, the rough endoplasmic reticulum, as well as the smooth endoplasmic reticulum. Remember to draw and label the basic organelles like the mitochondria, the smooth endoplasmic reticulum, and the rough endoplasmic reticulum. Remember that ribosomes can be 70S. These are mainly in prokaryotes, but they can also be found in mitochondria and chloroplasts of plant cells. They can also be 80As, which are mainly found in eukaryotes. Remember that centrioles are tubular. I have seen a previous question where students were asked to observe if these were centrioles or not, so please look at that. Remember tiny basics about the endosymbiosis theory. They've also asked that as well. Remember that lysosomes contain lysozymes, which are lytic enzymes useful in various cellular processes, including apoptosis, as well as destruction of old organelles. Remember to describe the function of the rough endoplasmic reticulum and the Golgi apparatus in protein modification and packaging. Remember the role of the smooth endoplasmic reticulum in lipid and steroid production as well as transportation. Please, you need to revisit Figure B on page 159. And this page demonstrates the rough endoplasmic reticulum and the Golgi apparatus and the involvement of the Golgi apparatus in receiving, packaging, and transport of proteins by exocytosis. Moving on to prokaryotic cells, please learn to draw and label well the bacterial cell, including all the major components inside, and remember the functions of the components of the bacterial cell, including the 70S ribosomes as well as plasmids. Remember the difference between gram-positive and gram-negative bacteria in terms of being stained. This question is rare and I don't think it will be asked, but you never know, so please do not ignore it. Going to classification of bacteria, they can be classified based on their shape. They could be rod, they could be comma-shaped, or they could be spherical, and so on. Also, they could be classified based on their respiratory requirements, like being obligate aerobics, facultative anaerobes, as well as obligate anaerobes. Cells can be arranged into tissues, as well as those tissues being arranged into organs and organs into organ systems. So on this note, I would like you to visit page 163 and look at Figure A and B, which talk about the epithelial tissues, as well as their functions. This question is also rare, but please do not ignore it. Going on to the cell cycle, remember the difference between sexual reproduction and asexual reproduction. Sexual reproduction is carried out through exchange of gametes, and these gametes are produced through the process of meiosis. Asexual reproduction does not require gamete formation or exchange, and cells go through mitosis in order to divide and increase in number. You need to remember that DNA is wrapped on histones, and then coiling occurs so that the chromosomes can be formed. Remember that a karyotype represents all chromosomes found within a cell. When we move to interphase, you need to remember all the three stages or all phases of interphase. There is the G1 phase where the cells take in material, grow and increase in size. Then we have the S phase where the chromosomes replicate, forming double-stranded chromatids. And we have the G2 phase where organelles and other molecules required for cell division are synthesized. So please make sure you fully understand what happens in interphase. And after that, we go to mitosis. We need to remember the changes that occur in every stage of mitosis, like in prophase, metaphase, anaphase, as well as telophase. You need to know the details of how the chromosomes are going to be moving, how they're going to be separated, and where they end up. You need to remember that cytokinesis in plant cells occurs differently from the way it occurs in animal cells. And a question could be asked to contrast or compare and contrast the two. We need to know the advantages of mitosis in terms of producing genetically identical offsprings. If we need organisms that are genetically identical produced within a shorter period of time and in large numbers, mitosis can be beneficial. However, there is a problem due to lack of genetic variation. So if there is any problem or any changes in conditions, there is a chance that all these are going to be destroyed. So you need to know the disadvantage. You need to remember how to calculate the mitotic index, and you should be able to observe images of different cells in different stages of mitosis so that you can use that to calculate the mitotic index. Going on to meiosis and sexual reproduction, you should know that sexual reproduction produces diploid gametes through meiosis, and when the male and female gametes fuse, a zygote is produced. You need to learn the details of meiosis, including meiosis 1 and meiosis 2. You also need to fully know how the contents of the cell change through each stage in prophase 1, metaphase 1, anaphase 1, telophase 1, as well as prophase 2, metaphase 2, anaphase 2, and telophase 2. Actually, a question could be asked to compare and contrast mitosis and meiosis. Remember the differences between these two. Please remember that crossing over occurs in prophase 1 of meiosis, and random assortment, or sometimes called independent assortment, occurs in metaphase 1 of meiosis. This question is very common. You need to know that these two processes, crossing over as well as random assortment, are responsible or partly responsible for the great variation that occurs in offsprings. However, there is another factor which is called fertilization. We know that fertilization occurs by chance, so due to that, there is greater genetic variation. You need to remember to draw and label the male gamete, which is a sperm, including the acrosome, the middle piece containing mitochondria, as well as the tail, and you should know the functions of each component of this structure. You need to revisit gametogenesis in plants, like the sporophyte as well as the gametophyte generation, although this question is very rare. You need to know the details of fertilization in animals, including the cortical reactions and their role in preventing polyspermy. This question is very common, so please ensure you fully understand those details. You are required to know the details of double fertilization in plants, including the role of the pollen tube nucleus, the generative nucleus in forming the two male nuclei, the role of the antipodal cells, and the role of the formed endosperm within the seed that is produced. Moving on to cell differentiation, you need to understand differentiation like alleles, codominance, and the ABO blood groups. You need to practice genetic crosses like dihybrid inheritance, although these are very rare. They could ask you about interactions between genes and the environment. These questions are mostly linked to data that you have to study and make conclusions. You could make suggestions, comment on, describe, or explain. So you need to fully understand the question before you attempt to answer. They could bring data on skin color, health conditions, or ask you to comment on how the changes in the environmental conditions have influenced the results. This is linking to the factors we saw above there. So remember that error bars or standard deviation, the number of studies that were carried out, these are going to be very key in determining whether the data is valid or not. Twin studies are commonly carried out to study these effects because the differences in data can only be due to the environmental changes and not due to genetics. You need to know the difference between continuous, like height, body size, skin and hair color, and discontinuous variation, like gender, blood type, and so on. Moving on to controlling gene expression, you need to remember that transcription factors are proteins that bind to DNA and affect the transcription process. On this note, please revisit the process of protein synthesis. Remember the details of post-transcription modification, like RNA splicing by spliceosomes to produce the mature RNA as the introns are removed. You need to remember DNA methylation and silencing of genes, as well as demethylation for removal of methyl groups. Remember the details of histone modification, histone acetylation, meaning adding acetyl groups to open and activate the chromatin. You need to know about histone methylation by adding methyl groups to cause inactivation. These are all contained in how genes can be expressed. You need to remember that these epigenetic controls can lead to cell differentiation. Going on to stem cells, and these questions here are very common. Remember, stem cells can be totipotent, like the zygote. They can be pluripotent as well as multipotent. I included that the zygote is totipotent, while the blastocyst contains pluripotent stem cells. You need to remember that as organisms grow, gene expression, which is switching on or switching off of genes, can be achieved. through epigenetic control mechanisms like DNA methylation, histone modification, as well as the use of transcription factors. You also need to remember the examples of fetal and adult hemoglobin on page 211. Moving on to stem cells, questions can be asked about stem cells and therapeutic cloning. You need to remember the description in figure B on page 213 about that. You need to learn about stem cell therapy, its applicability, the advantages as well as disadvantages, and you need to know about using stem cells and case studies with experimental results like diabetes, Parkinson's disease, as well as other genetic diseases. You need to know about using stem cells and the ethical issues surrounding them. And finally, the advantages and disadvantages of using induced pluripotent stem cells over fetal stem cells. Topic 4, plant structure, function, biodiversity, and conservation. I began with plant and animal cells. You need to learn how to draw and label the structure of an animal and plant cell. Remember the functions of the major components. They can ask you to compare and contrast between the structure of the plant and animal cells. Please remember chloroplasts, the permanent vacuole, as well as the cell wall. These are components found in the plant cell that are absent in the animal cell. You need to remember the major components of the plant cell wall and their function, components in the primary as well as the secondary cell wall. You need to learn to compare and contrast the primary as well as secondary cell wall, and this question is very common. Moving on to cellulose, you need to remember that cellulose contains beta-glucose molecules and it contains only 1,4-glycosidic bond, unlike starch. You need to remember there is inversion of some beta-glucose molecules as the 1,4-glycosidic bonds form between the two glucose molecules. However, cellulose molecules will remain very long straight chains, unlike starch. So it is not suitable as a storage molecule, but good as a structure molecule. They can ask you to compare and contrast the structure of starch as well as cellulose. And on this note, they could ask you to compare and contrast the structure of glycogen as well as that of cellulose. They could also ask you to compare and contrast the structure or function of cellulose and other molecules like the protein collagen and so on. Remember the importance of the plasmodesmatter. It is to allow communication between cells. When we go to plant organelles, details of the structure and function of the vacuole could be asked. They can ask you to draw and label the chloroplast. Remember to include the outer membrane, the inner membrane, the thylakoids, starch grains. Remember this is big. Include the grana as well as some ribosomes and genetic material. Remember that amyloplasts store starch. Moving on to plant stems, remember about plant tissues that make up the stem, like the collenchyma, sclerenchyma, parenchyma, xylem, as well as the phloem. Please revisit all the details, including how to identify these structures from images. Previously, questions have come where images have been presented to students and they were asked to label those structures, so ensure you fully understand how to identify them from a specific image. Remember that the function of xylem is for water as well as mineral ion transport. Remember that mature xylem are dead lignified tissues and these vessels are arranged vertically within the stems. You need to remember that the phloem carry out translocation of substances like sucrose and they have companion cells that control the processes that occur inside those cells. Also, you need to remember the role of the following mineral ions to plant tissues. Remember that nitrates are used for production of amino acids that can be used for making proteins. Calcium is useful in making calcium pectate, which is found in the middle lamellar, and magnesium is useful in making chlorophyll. Moving on to plant fibers, here different information could be given as data, and you could be asked about uses, mainly in terms of strength. They could ask you the uses of bioplastics, their advantages as well as their disadvantages. On this note, they could also give you images of plant fibers and ask you to compare and contrast them in terms of their durability, their strength, as well as their properties as useful materials. Moving on to plant-based medicines, remember to use aseptic methods for bacteria growth and culture. They could ask you questions about response of microorganisms to antimicrobials. This question could be common in Unit 3, but it could come in Unit 2 as well. They can ask about extraction of components from plant materials as described on page 239 in your textbook, so please ensure you fully understand. A question could come on how they can use plant material to develop new drugs, and they could ask you which steps the scientists should take in order to make this drug available. That means they want to talk about clinical trials. So you need to understand that these are carried out to ensure that the drugs are safe, they are efficient, they are stable in the body, and so on. Also, drugs should be affordable and easily produced on large scale. So please remember all phases of clinical trial and their importance, as well as the need for a blind trial. This is a very common question. You should fully understand it before you go for any Unit 2 biology exam. Moving on to classification, remember to distinguish between eukarya, archaea, as well as bacteria. Here I use the three kingdom system. Remember that eukarya include animal, the fungi, protista, as well as eubacteria. Remember methods used for classification include, they can use morphological appearance, they could use the ability to produce fertile offsprings, as well as similar molecules using genetics. You need to remember that DNA profiling can be used to tell if two species can be classified in the same group based on the similarities they possess. Remember the basics of the endosymbiotic theory. Moving on to biodiversity, this topic involves a lot of data, and the data could come in form of graphs, in form of images, in form of tables, and so on. So please try your best to fully understand the question before you attempt to answer. They could bring definitions like what is biodiversity, what are ecosystems, what is species richness, what is abundance, and so on. Remember to make sure you fully understand those definitions. They could also bring questions relating to endemism. They can ask factors that affect biodiversity in a specific area. Please ensure you fully understand. They could bring data where a specific change has occurred, and they ask you to evaluate or to explain the change on the biodiversity. Ensure you observe the data, observe which change that has occurred and how it has affected the organisms within a specific area so that you make a conclusion or so that you give an explanation for the changes. You need to remember how to calculate diversity index. This formula is usually given in the exam, but you can use page 270 in order to practice for that part. On measuring biodiversity, they could ask about using the heterozygosity index. Please remember this is the number of heterozygotes divided by the total number in the population. The next part could be adaptation to a niche. They could ask what is a niche, what is a population, allele frequency, and so on. They could also ask about anatomical, behavior, as well as physiological adaptations. You must be able to look at images, organisms, and extra given data to determine the adaptations that enable organisms to survive in a specific area. Examples include the desert where the temperatures are really high, or the North Pole and polar bears where the temperatures are really cold. This is a very common question, so ensure you fully understand how to reason that. Next, they could ask about the Hardy-Weinberg equilibrium calculations. Please visit page 278 of your textbook to fully practice. They could ask about the effects of isolation on gene flow between two populations. When we come to speciation, they could ask about allopatric and sympatric speciation. They can give various images and ask questions about how organisms became two different species, yet they had a common ancestor. Moving on to conservation, they can ask about conservation in animals like captive breeding programs in zoos as well as wildlife parks and their importance on conservation. Remember to describe how captive breeding is carried out. You should know what should be done before the organisms are reintroduced. For example, they need to educate the natives. They need to make sure the conditions that caused decrease in the number of the species are dealt with before the species can be introduced within the specific area. They can ask about the advantages and disadvantages of captive breeding. And when we go to conservation in plants, they could talk about seed banks and how seeds are prepared for storage before they are kept in a seed bank. This question is very common, the question on conservation. You have to make sure you read about seed banks as well as conservation in animals. Please revisit methods for calculating standard deviation on page 297 under the math skills. In addition to the topics we have seen, please remember that for any biology question, describe means how something happens. When they ask to explain, it means why something happened. So here you need to give reasons as to why something happened. And for longer questions, some points from your own knowledge could be added as well because they could give you marks, provided they are related to what you're trying to talk about. When they ask you to compare and contrast, they want you to talk about the similarities and the differences. And when they say discuss, they want you to find the issue or situation raised in the question and then write a detailed analysis of that situation. Lastly, when they say comment, they want you to get information from the given data and make a judgment about that piece of information. So this brings us to the end of these potential questions that could be asked in any Biology Unit 2 paper. Thank you for being with us. Please do not forget to subscribe. See you in the next video. Bye-bye.